the interest in starting right on time, we'll get going with our second ever INDIGO seminar and our first seminar of, I'm sure, more in the future on Alzheimer's disease. I'm so pleased that Dr. Albert Gonzales and Dr. William Torch can join us and share their expertise with us. Dr. Gonzales, is this your first semester at UNR?
Yeah, I started in October of last year.
Okay, very exciting. Well, we're so pleased to have you. Just for a brief introduction, you're coming to us from University of Vermont previously, is that right?
Yes.
Okay, I have noticed that you did your undergraduate at California State Polytechnic and got your master's in biological science there as well. Did your PhD in biomedical science and vascular physiology at Colorado State University in Fort Collins, and a postdoc there as well in biomedical science. And so you were last in the Department of Pharmacology at UVM before coming to UNR?
Yep.
Right. And it looks like you have a really recent publication on PIP2, improving cerebral blood flow in mouse model of Alzheimer's disease, that just was published this month.
Yeah, I think yesterday or 2 days.
Yesterday?
Yeah.
Wow. So this is very new and exciting information. Is there anything I'm missing?
No.
Okay. Well, with that very brief introduction, we'd love to see your presentation.
Thank you. Let's see if I can do the share screen. Okay. Do you guys see my title slide?
Yes.
Yes. All right. I want to thank Mary for giving me this opportunity. I'm new to the University of Nevada, Reno. I started just October of last year. Starting up a lab during these interesting times is interesting by itself. So I want to talk about some of the work I did prior to coming to UNR, looking at amyloid beta as a part of Alzheimer's disease. Here's some information about me because I'm new. I'm in the Department of Physiology, Cell Biology. Here's my email address. And if you're interested, I actually put up a website, gonzaleslab.com, where, just showing the general what we were interested to do in the lab. So just wanted to do a quick outline of what the seminar was going to be. First, I'm going to talk about some of the old work we published, looking at how blood flow in the brain is unique. And then try to define some key aspects of blood flow regulation in the brain in the sense of capillary-mediated functional hyperemia. Recent publications from my lab from 2017 showed that the capillary network is actually the sensory network capable of sensing tissue metabolic activity and responding by increasing blood flow. And then the second paper I'm going to quickly go over is this role of these novel, pseudo-novel cell types, contractile pericytes, and how they increase the regulation of capillary blood flow. And then I'll come bring it back and actually show how within the disease state, amyloid beta can have a profound effect on these contractile pericytes that regulate capillary blood flow. And so the first idea I want to get across is the purpose of the vasculature. The vasculature must meet the metabolic need of the tissue. And this is especially important in the brain, or feeding the brain, where the brain constitutes only about 2% of the total body weight, but it consumes about 20% of the glucose reserves. And this is important because neurons have a high metabolic demand, but lack any kind of energy reserve, requiring a constant blood flow. Although this is really important, and this is just the basis of a lot of work, I want to make an argument that that is not what makes the brain unique. Mainly because other tissues are more metabolically demanding. For example, the heart, which makes up a smaller fraction of the body, constitutes almost a third of the body's energy, and the kidney is the same. So you have other tissues that are just as metabolically demanding, But what makes the brain unique is this process. When you're processing information in the brain, different neural circuits or groups of neurons are engaged, leading to scenarios in which groups of cells neighboring each other may have a different level of activity and exhibit a different metabolic demand. So this heterogeneity of the cellular activity leads to a heterogeneity in the metabolic demand. The argument I would make in the heart is extremely metabolically demanding, but during contraction relaxation, majority of the cells are pretty much doing the same thing. Unlike in the brain, as you process information, groups of cells become active while neighboring cells may not. And therefore, the brain requires a finer control of the delivery of oxygen and nutrients, and in particular, in regards to Alzheimer's disease, the removal of waste products. And so recently, my postdoc lab came up with two publications that sort of highlighted some of the new roles of the capillary network in functional hyperemia or neurovascular coupling. This first paper that came out, I said, in 2017, sort of highlighted the capillaries as a sensory network responding to the metabolic needs of the brain. And this is done simply by, during neural activity, one of the byproducts of neural activity is the release of potassium. This potassium can activate KIR channels within the capillaries and generate a hyperpolarization. This hyperpolarization, because of the unique qualities of this ion channel, can propagate retrogradely or towards the arterioles to lead to a hyperpolarization and relaxation of the arterioles. This relaxation of the arterioles leads to an increase in blood flow that conducts itself downstream to feed the neighboring or active neurons. To add to this, recent paper we published last year in PNAS looked at the novel role of contractile pericytes. So typically, when you look at the vasculature, you have your arterioles that give way to capillaries, which is the direct site of exchange between the tissue. And within the capillaries, you have a different type of mural cell called pericytes. These pericytes are found throughout the body in almost every vascular bed, and they pretty much cover the whole capillary network. But like other cells, pericytes actually have subdivisions, different morphologies, different functional contributions. And so in this paper, we focused on pericytes localized right in the beginning of the capillary network, where blood entering the capillary network could be influenced by these cells. And what we showed in this paper is that these cells localized at capillary bifurcations were actually capable of deviating blood, controlling which direction blood flow went, whether it went down one branch or another branch. And so these two, papers together, we sort of came up with this concept of capillary-mediated functional hyperemia, where in summary for both papers, when you have neuronal activity, that neuronal activity generates a retrograde hyperpolarization, an electrical signal that propagates up towards the arteriole. This electrical signal causes the arteriole to dilate and increase blood flow. But because of the pericytes, which are localized in the capillary vessels in the same path of the electrical signal, they too can receive the electrical signal, dilate a specific branch to ensure that blood flow follows and is directed to where the active neurons are. And so, we developed this idea more for the delivery of oxygen and nutrients to the active neurons, but we now can propose whether this mechanism is important for waste removal. And in particular, we looked at the model of Alzheimer's disease, and in particular, amyloid beta. So just a really super simplified overview of the role of amyloid beta in Alzheimer's disease is on neurons and endothelial cells, you have these proteins called amyloid precursor proteins. These amyloid precursor proteins get cleaved by these secretases, um, which leads to the generation of these amyloid beta oligomers. And typically, under normal conditions, these peptides are pretty cleared out pretty quickly by either being taken up by microglia or just being directly absorbed by the circulation. And so when the problems start to arise when there's a breakdown in this clearance, this ability to clear these amyloid beta proteins. And so the amyloid beta starts to aggregate. And so you start to form amyloid beta oligomers, fibrils, and eventually plaques. And that these plaques cause, a systematic, degradation of the neural network. And so for our research, we wanted to see what role could amyloid beta play in the circulatory system. Is there a direct role of amyloid beta on the blood vessels and their role in clearing the waste material? And so to look at these questions, to address these questions, we used a transgenic mouse driven by smooth muscle myosin heavy chain, driving the expression of this fast calcium indicator so we can look at calcium dynamics, these change in fluorescence. And we decided to use the retinal preparation as a high throughput ability to visualize the almost complete vasculature. And so here's a typical experiment. Just to show you example of what's happening. We're just going to place on 60 millimolar potassium, a very high concentration of potassium. And this high concentration of potassium collapses the potassium gradient of the tissue, which causes membrane depolarization. Membrane depolarization leads to activation of voltage-gated calcium channels, increases cytosolic calcium, and you get a sustained vessel constriction. And so here, an example movie, on top up here is a feeding arteriole, and coming down is the pericyte-covered capillaries. And so when we play the movie, we put on 60K, we cause membrane depolarization. Hopefully you can see the fluorescence goes up, you get a brighter image, that means more calcium, and you get this sustained constriction. Okay? So on this prep, we wanted to see what happens when we put amyloid beta on. First of all, we can summarize what we see here. Looking at the arterioles, looking at what happens when we put 60K, we get an increase, more calcium events. And same with the pericytes. We see an increase in calcium events. And also, the increase in calcium events leads to a constriction. So if you looked at the diameter of the arteriole, the diameter decreases or constricts in the presence of 60 millimolar potassium. The same with the pericytes. These pericytes constrict in the presence of the 60 millimolar potassium. And so now we wanted to see what amyloid beta does. So you can purchase this amyloid beta peptide, and in this case we're using 1 to 40 the amino acids 1 to 40 of the amyloid beta, and you can pop it on the prep. Here we have the feeding arteriole on one side, and then we have these these pericyte covered capillaries. And then when we put it on, hopefully you can see it, you get this increase in calcium events. You get more little calcium events occurring within the pericytes, within this region here. But if you looked at the arteriole, not much happened. And so we can summarize that here, where we did not see an increase in calcium events in the arteriole, but we did see an increase in calcium events in the pericytes. And this increase in calcium events, when we looked at the vessel diameter, didn't change the vessel diameter. So we just got a bunch of calcium events. But what was really interesting is what happened afterwards. And so in this experiment, we put the amyloid beta on. We get an increase in calcium events in the pericytes, But then we followed up with 60 millimolar potassium. So if we remember back, just putting on 60 millimolar potassium constricted everything. But let's see what happens in this prep. Again, down the middle is the feeding arterial, and all these little side branches are the pericyte-covered capillaries. So when we put 60 millimolar potassium, you can see an increase in the fluorescence. Fluorescence goes up really high, and you get a constriction. But what was really interesting is that the constriction or response to 60 millimolar potassium only occurred within the arterioles. The pericytes, the side branches here, didn't respond to 60 millimolar potassium. And if you track the diameter of the vessels, in the presence of amyloid beta, we were still able to get a constriction of the arteriole, but the pericytes, the capillaries that the pericytes are on, didn't respond. And so we came to a couple small conclusions. So that amyloid beta is selectively stimulating calcium events within the capillary pericytes. That the amyloid beta induced calcium events do not stimulate a contraction in the pericytes, and that the amyloid beta calcium events, and we're still investigating this, prevented the membrane depolarization induced constriction of the capillary pericytes. And so in a short conclusion in this, that amyloid beta peptide mediated this increase in calcium events, which we're going to term short circuits the pericyte calcium signaling and contraction through the inhibition of voltage-gated calcium channels. But this was through the delivery of amyloid beta. We wanted to look at if there in the mouse model, can we recapitulate what we saw of this loss of contraction in pericytes? And so to address these questions, we used a mouse model for Alzheimer's disease, in particular amyloid beta doses. These 5X FAD transgenic mice. These mice express 5 of the familiar Alzheimer's disease-linked mutations. And through the progression of their life, they exhibit a lot of the Alzheimer's symptoms. In particular, you get this extracellular amyloid beta deposition, so you get all the amyloid beta clusters here. And so here's an image of a 5X FAD mouse. In red is the blood vessels, so we inject the mouse with a fluorescent dye so you can see all the blood vessels. And then this methoxy-X04 actually labels all the amyloid beta in the brain. So in the brain, you can see a lot of these little puncta of fluorescence, which is the amyloid beta. In particular, you can see on the arteriole here, there's a lot of aggregates of amyloid beta. And as the mouse progresses to 12 months of age, you get both cognitive impairment and neuronal loss within the cortical layer. So using this mouse and returning back to the retina, we wanted to see what happens to our 60K response in these mice. And so in the retina itself, we can also see amyloid beta plaques using this methoxy dye. You can see the little yellow puncta. And so again, here is baseline. This is the 5X FAD mouse. Here's the arteriole, and here's a branching off capillary with the pericytes on them. When we put 60 millimolar potassium to cause membrane depolarization, hopefully you can see and appreciate here that the arteriole does constrict. You get a decrease in the diameter of the arteriole, but in these mice, the capillaries or the contractile pericytes did not respond. If you looked at the cross section here. Now, we were able to rescue this effect using this drug, and this drug is not really specific. It requires more work, but this drug is this SKF 96365 can block a lot of store-operated calcium entry and transient receptor or TRP channels. And when we put that on in these 5X FAD mice, we can cause a constriction of these capillary pericytes, rescuing the effect that we had before. Um, so, again, a short conclusion here. Again, amyloid beta, we propose or is playing a role in short-circuiting pericytes, specifically pericytes, and their role in the calcium signaling and their ability to contract. And we still have to do some more work. This is still very preliminary results, um, where we have to look at whether we know that pericytes are being disrupted. Is their role in this directional control of blood flow affected in these mice, in sense that when you have neuronal activity, that retrograde hypopolarization can go upstream, but the pericytes do not respond to it. And also very importantly, we have to figure out why amyloid beta is specifically affecting pericytes, these cells within the capillary network, versus the feeding arteriole, the larger vessels. And so we still have a lot of observations to iron out, but this is a work in progress. So just an acknowledgement. I'd like to thank the Nelson Lab, where I did my postdoc in Vermont, and as a new faculty here and growing lab here, I would like to thank the lab here, Ahmed and Christian, getting things started. As you can tell, we have empty shelves here. Just needs more and more equipment. Again, if you have any interest and you want to see pretty movies, you can come to the website gonzaleslab.com. And of course, I want to thank the funding that we get. And thank you. Any questions? 20 minutes.
Thank you so much. That was great. And you had a lot of pretty videos, so we got a few of those. We do have time for a few questions if anyone has a quick question. My question is, it seems like the amyloid beta is messing with waste removal and dysregulating the blood flow. Is that like a fair typification of what you're seeing?
Yeah, so there's always been sort of a correlation between cardiovascular disease and the onsets of Alzheimer's disease, as in you're perhaps more likely to have Alzheimer's if you have some cardiovascular disruption. So the idea here is placing the vasculature in this unique role in removing the waste product that could be generating even prior to the amyloid beta accumulation or aggregation. And the idea that without the proper removal of not just amyloid beta, but other waste products, you can lead to an environment, a tissue environment where they're just not optimal for tissue function. And so, everything that's happening within the neurons and the brains could still be happening, of course, but it's the role of the vasculature to help prevent that. If that makes any sense.
Yeah, thank you.
Where do you think the beta amyloid is coming from? What is the source of the beta amyloid? And teleologically speaking, why would the body want to do that?
Yeah, so you bring up a really good question. A lot of the focus right now is, and especially within the vasculature, is this effective amyloid beta. Amyloid beta does this, does that. But like I presented in the beginning, it's a breakdown and then its actual clearance that causes the aggregation of amyloid beta. And so we are starting to look into mechanisms that breaks down behind the scenes, breaks down the clearance of a lot of waste products within the brain. And for the role of amyloid beta, I started to look into it because before I've sort of just said it was a surface protein on a lot of neurons and endothelial cells. But I'm starting to look at the role of amyloid beta as contributor to iron transport. There's a lot of literature looking at iron, the role of this APP, the amyloid beta precursor protein, in iron homeostasis. And so that's the direction we're sort of going. But we don't have anything really to present right now, but soon we'll have a bigger story about that. And so if we can address, what's happening before the aggregation of amyloid beta, before the disease, our ability to visualize the disease, we can see this, a breakdown that's happening behind, or in the dark, as a sense.
Thank you. Do we have a couple more questions, or Dr. Torch, do you want to start getting your presentation open, and anyone's free to ask questions?
Yeah, let me do that. Can I just go to share screen?
Wait, I have to give up my screen.
Oh, I gave you up already. You're okay.
Oh, okay.
And does Dr. Kidd want to say anything? I didn't want to cut in front of you, Dr. Kidd. Did you want to comment, or?
I had a question. It looks like we're moving on. I mean, if you want to, I'm just curious. What do you think's killing people or neurons in Alzheimer's? Is it a lack of blood supply?
My talk will give the answer.
Let's get an unbiased response then.
I mean, the idea is that the neurons, you get a degradation of your neuronal, and you get fewer neurons, and that leads to a lot of more detrimental things. But what I'm trying to show more is what's happening way before you get the huge amyloid beta plaques, or the actual neuronal cell death, is that there's a dysfunction happening within the vasculature. Something that's not really paid attention to too much, and as the end result. Yeah,
and I just want to slide in a quick question. I'm a second-year medical student, so I care about drugs, right? And I want to know more about the treatment options. So currently, I was just looking at what do we use to treat Alzheimer dementia, and it's acetylcholine esterase inhibitors, which sounds like, after I did a little bit more reading, basically the neurons are dying, the cholinergic neurons are dying. And so we're just trying to give medication that increases the concentration of acetylcholine in the brain, so you don't get all those symptoms. It's symptom management. I was wondering, are there other medications that you know of, or other targets that might not just be symptomatic treatment, but as you're saying, that address the blood flow, that either address the production or elimination of these beta amyloid and tau proteins?
Yeah, I mean, they're starting to address this by looking at a lot of the drugs that to treat cardiovascular health, and seeing the effects of the progression of the disease, of Alzheimer's disease, and it's promising. It's helpful, but again, we are still just understanding how blood flow is controlled in the brain. At the finite level, at the cell to cell different level. And the hopes with our work is to be able to do. One reason why I'm looking in the retina is to be able to help in the prognosis, or identifying Alzheimer's disease earlier on, as a non-invasive way of looking at the vasculature of the brain, and in hopes of when these pericytes are starting to die off, does that occur prior to the formation of the larger amyloid beta plaques? And so you can sort of diagnose a little earlier, and then treat, hopefully, way before you're getting all these neuronal cell deaths or such.
Now, Biogen has come out with a proposed drug which they claimed was going to help clean up or remove the amyloid deposits in Alzheimer's disease. And I understand that a review of their claims denies the fact that their drug can actually do the job, so to speak. Are there any other drugs in the pipeline that might be used in that fashion? If beta amyloid is the answer, the removal of beta amyloid is the answer to reversing the consequences that produce Alzheimer's disease.
Yeah. So, I don't like to say this, but amyloid beta is not the only problem when the disease progression. And so this is one reason why I'm trying to take a step back, further back, and not just look at amyloid beta, but looking at the problem of just clearance of waste products in the brain. This is applicable to, especially with, the tau protein, the role of tau in forming the tangles and such. So once we take it back a little further, then we can start to approach, different therapeutic targets to help ensure that the health of these pericytes, these cells that control capillary blood flow, are maintained, and their role in especially the clearance of waste. So no, not yet.
All right.
To keep it simple. But there are a lot of promising ideas linking, treating cardiovascular disease and seeing the effects on neurodegeneration.
What I find most fascinating are the capillary and precapillary arteriolar changes that take place as a result of high potassium and calcium effects in the vicinity of those blood vessels. It's almost like a faucet that's being opened and closed to allow blood flow with oxygen to get to the areas that are somehow self-modulated, at the local level, cellular level, either local or even downstream, which then affects the neuronal functions, permitting directional electrical conduction down appropriate pathways.
Mm-hmm.
I find that absolutely fascinating. It's a beautiful model. And when I look at amyloid, I think of my sink getting gunked up with stuff that's gone down into the pipes. and you have to take a Roto-Rooter to clean it out. I know that's a gross kind of thought, but anyway, at that level.
I can actually also flip that, the important role of feeding, the role of the capillaries to send blood and oxygen to neurons that are active is important. But what happens when there's disruption or a breakdown and blood is being sent to inactive neurons? Does that increase in oxygen and nutrients lead to a spontaneous increase in activity in those neurons?
Right.
You can start to, we can really start messing up with the brain and the neural circuitry just by messing up the vasculature.
Right. Wonderful
Dr.
talk.
Torch, are you ready to give your presentation?
Yeah, should I just go to share screen?
Yeah.
All right, let me do that. And then I have to click on this one.
Beautiful.
It works? Okay.
It works.
You can see my cursor?
Yes.
Okay, thank you, Marissa. Thank you, Mary.
Yes.
I couldn't have done this without you. I'm going to be talking about a project that has involved my entire professional life for the last 50 years. And I've given this talk to at least 20 different universities and meetings. And I've been advised by many people who've listened to this talk that I should call this a unified grand theory model for progressive encephalopathy leading to dementia, behavioral, sleep, and autonomic disturbance following hippocampal injuries. So the talk I'm going to be given is entitled Progressive Limbic Anterograde Transsynaptic and Transneuronal Degeneration in Diseases Presenting with Alzheimer's Dementia-like Symptomatology. There we go. As far as my background, I'm a board certified child neurologist specialist, also with competence in sleep disorders medicine. I've run the Washoe Sleep Disorder Center for the last 15 years. I'm now retired, as well as the Neurodevelopmental and Neurodiagnostic Center, where I saw adults and children with all kinds of neurological conditions due to stroke, trauma, encephalitis, et cetera, et cetera. I've seen thousands and thousands of patients. I was the former director for the Northern Nevada Muscular Dystrophy Association, where I saw lots of ALS patients who had dementia on top of underlying ALS issues. I just retired. I'm an inventor on the side, have about 50 patents relating to eye tracking and EEG, which might be of interest to the department. As far as my background training, I got my bachelor's degree in chemistry at Brooklyn College, my MD degree with honors in research at the University of Rochester School of Medicine, where I got my MS at the Center for Brain Research and in the Department of Biochemistry. I did my pediatric internship at Mass General Hospital, a child psychiatry residency there as well. I joined Lou Sokolov, who is the editor of the Journal of Neurochemistry, and worked as a research associate in the lab of cerebral metabolism at the NIMH, and then came back to finish my pediatric and neurology residencies at Albert Einstein, where my interest in limbic degeneration began. I've developed this grand theory. I hesitate to call it grand theory, but I'm going to use it because others have said I should. And I'm going to be talking about what happens following hippocampal injuries of any kind, whether they're vascular, infectious, traumatic, metabolic, or neurodegenerative, as in Alzheimer's disease and a list of many other dementias. And in the process of talking, I'm going to bring up the fact that while this has gone on for 50 years, I can now address what is happening in COVID-19 in terms of the relevance to what is happening in today's world. So the question that I'm asking is very provocative. What if it's not Alzheimer's disease? What if Alzheimer's disease really is a multiplicity of many different diseases that produce an Alzheimer's-like syndrome? And what if limbic anterograde transsynaptic degeneration is the basic mechanism that operates in those 20 to 50 different diseases? I've given this talk at many different venues, and I'm just going to show just two big slides on the neuropathological histological biomarkers that can be seen in the limbic system following post-mortem evaluations of people who've died of different diseases where the hippocampal formation has been injured. I've also been looking at biological markers that could be used in living individuals, perhaps to be studied neuroradiologically, and I'm going to go into those findings. The case started when I was a resident. I was around 30-something years old, and I was working with Dr. Hirano, who was the head neuropathologist at the University of at the Albert Einstein University. He was the discoverer of Hirano bodies, which, like Lewy bodies, was key to identifying Alzheimer's disease back in the early 1970s. Dr. Solomon was my attending neurologist who took care of the 64-year-old gentleman whose history I'm going to describe next. The patient who I was asked to review as a resident was in great health before the age of 56, and over the next 8 years developed a progressive dementia of unclear etiology. Early in the stages of his dementing process, he became childish, irritable. He developed paranoid, apathetic, introverted behaviors. He was sexually impotent and depressed, and there was progressive mental deterioration with advancing short and long-term memory loss. By age 60, he had end-stage dementia, and he was totally dependent. He continued to live onwards. At age 63 and 6 months, he had a circadian rhythm disorder where he was sleepy during the day and it had insomnia at night. He dropped over 50 pounds. By 63 years and 6 months, he was severely demented. And what was most peculiar, unlike in Alzheimer's disease, there was a large left temporal lobe EEG slow wave focus that no one could understand. Now, mind you, this was before MRI and CT scans were invented. I was one of the first people to see the first CAT scan in 1977 when Albert Einstein built their first machine. So we had no idea what was going on in this man's brain. He also had a left-sided Horner syndrome, which involved droopiness of the eyelid and pupillary changes. He had variations in his blood pressure, a dysautonomia. He started urinating. He was drinking a lot of liquid and had uncontrolled bladder control, gastric and pulmonary secretions. He lapsed into coma, developed tachypnea, tachycardia, hyperventilation, and he died of a cardiovascular arrest. Dr. Hirano, who was his attending doctor, attended the Grand Rounds conference where I presented the autopsy findings that Dr. Hirano had raised and was astounded by. And we asked Dr. Solomon to stand up in front of the audience to say what he thought his patient had died of. He says, oh, clear cut, multi-infarct dementia, possibly Alzheimer's disease. Wrong. At the autopsy, whoops, let me go back here. Here's what was seen. His brain was normal. He did not have Alzheimer's plaques, tangles. There were no Hirano bodies. All he had was this big left hippocampal infarct, about the size of a plum. And here's the cross section. Everything is reversed. This is really his left brain. You can see the hippocampus, the parahippocampal gyrus, the amygdala, all cystically infarcted. And we estimated this was 8 years old. It was scarred. This was the original abnormality. The rest of his brain was absolutely normal. But look at the ventricles, the third ventricle, the mammillary bodies, and let's get another picture. This is a picture of the limbic system from a 3D models. Here's the hippocampus giving rise to the fornix and fimbria into the mammillary body. Two little stalks that go up is the mammillothalamic tract into the thalamus that sits in the middle of the brain. And there's this is Papes circuit on the left, and there's an equivalent one on the right. This is the corpus callosum. This is a front view, and this is an interesting rear view. So getting back to what we saw at another level, let me see here. Here's Papes circuit. Here's the fornix on the normal right side. Here's the fornix on the left. It had shriveled to a wisp. Here's the mammillary body on the normal right side. It's gone on the left. Here's the mammillothalamic tract stained for myelin staining. Normal on the right, gone on the left. Here's the hypothalamus, fairly robust and thick in the lower part of the third ventricle, atrophic on the left. Here's the thalamus, nice and plump on the right, shriveled on the left. When Dr. Hirano looked at this, he said, I've never seen this before. And they said, Bill, you need to do a literature research and we need to write a paper. These are the areas in the hippocampus that were damaged. the presubiculum, subiculum, prosubiculum, dentate gyrus, alveolus, the hippocampal formation, and the fimbria fornix. This was all involved, and we presume was due to a posterior cerebral artery infarct, a vascular stroke, which damaged this area. But go back here. This is Page circuit. I did this thing about 40 years ago. Here's the hippocampus. Here's Page circuit, the fimbria fornix, going to the mammillary body, the mammillothalamic tract, the secondary neuronal pathways, and then the anterior thalamus projecting into the cingulate gyrus. Also fibers going to the prefrontal region, with fibers going down into the brainstem as well. Right? And this relates to ALS and also Parkinson's disease, which I will bring up. The limbic system on the left and the right control the orbitofrontal, or they integrate the hippocampus with the various parts of the brain, the orbitofrontal, dorsolateral, ventromedial, and cingulate portions of the brain that are involved in frontal executive functions. In order to appreciate what happened in that gentleman, I postulated that there were going to be maybe 5 stages to consider. And here's Page circuit. Here's a normal hippocampus and amygdala. Here's the fornix. Here's the mammillary body. Here's the mammillothalamic tract. And here's the projections into the cingulate and the frontal cortex regions of the brain. So we have primary and secondary and tertiary neurons that are trophically supporting each other in a domino-like effect that maintain functions of the limbic system in that hemisphere. In the first stages, you get a vascular injury that is local, as might have occurred when the gentleman had his stroke. The rest of the brain was normal. Stage C, progressive degeneration progressed from through primary and secondary neurons, leaving the cingulate gyrus intact, and stage D, further degeneration until the entire limbic system is wiped out. So I postulated that the stages of progressive LAT&D could involve denervation hypersensitivity, disinhibition, resulting in behavioral, affective, and psychomotor disturbances, including schizophrenic hallucinatory phenomena, and possibly complex partial seizures, which we see with hippocampal sclerosis. In the second stages, mammillary body and hypothalamic loss might result in not only progressive memory problems, but hypothalamic autonomic instability and sleep disturbances, which are involved in hypothalamic activity. And then progressive failure to thrive and a reduced survival time. Progressing then into the cingulate gyrus and the anterior thalamus, which can produce a limbic dementia. And it's known that strokes in the anterior thalamus by itself can produce profound dementia. Tumors in the anterior thalamus. So this staging is based on the reality of clinical progression in human mammals. And an extension of this hypothesis is that LAT&D may be an unrecognized generalized process that might be contributing as a factor in numerous progressive dementias and neuropsychiatric diseases, as I'm going to show next here. To test the hypothesis, I went back to the literature. I read a little French and German, and when I was in New York, I went to the New York Academy of Library of Medicine, and I found articles on the neuroanatomical and neuropathological findings that were written in the late 1800s and early to mid 1900s that described what was known as Pepys circuit, described by Pepys himself in 1937. I decided, well, we knew stroke can produce limbic anterogrades and degeneration, but what about other diseases? I went to Greenfield's neuropathology textbook, and I looked up vascular, infectious, trauma, toxic metabolic diseases, neoplastic, neurodegenerative conditions, including Alzheimer's and PICS, et cetera, paroxysmal post-epileptic encephalopathy, congenital, and other inborn errors, including autism in infancy, and found that in every one of these conditions, you could find limbic degeneration to some degree. To simplify this study, I decided to look at stroke, as in the patient, and found 49 cases, 24 cases of trauma. These were boxing injuries resulting in dementia precox, precocious dementia in boxers. Now we see it in football and chronic traumatic encephalopathy. Encephalitis, most of these cases were herpes encephalitis, had nothing to do with COVID. And then I decided to look at kernicterus, which is, as a pediatric neurologist, is nothing more than hyperbilirubinemia due to damage in the brain and breakdown in the circulation due to hypoxia and neonatal injuries that can cause mental retardation in children. I analyzed these over 130 cases and found that the mean age of onset correlated with the onset of diseases in that group. So the mean age for the 49 cases of stroke was 59 years of age, and these patients lived until the age 81. They had survival times that lasted anywhere from 2 months all the way to 21 years. They were equally divided into male and female, perhaps more male, more men than women that had stroke. Here's the boxing group, 24 gentlemen, average age 38, all men, of course. These were the boxers, no females. Herpes encephalitis, 41 cases, more men than females, and kernicterus equally distributed among children as far as sex. I decided to analyze those cases with my own methodology, just reading the descriptions of the neuropathologists. And I created a scale of microscopic and macroscopic. If the viewer said that it was visible to the naked eye, I called it macroscopic. If it was only seen under microscopic examination, I categorize it as mild, small x, XX, or severe, and the same at the macroscopic level. Here are the 44 cases of stroke lined up belonging to survival time. And it's absolutely fascinating that you could see, here's the limbic system, hippocampus, ventricular dilation in the lateral temporal lobe, mammillary body, hypothalamus, mammillothalamic tract, and you could see no mention of limbic degeneration in these cases down to here, but then after this point here, lots of abnormalities going into the thalamus, and when you look at the symptomatology associated with that, the pink areas are described by the authors themselves as dementia, when beforehand it were just behavioral changes. So this shows that with stroke, just as in our case, I can present the theory that limbic degeneration, whenever hippocampus is injured, can result in behavioral changes and end-stage dementia. The same thing goes for traumatic brain injury, dementia. No signs of dementia in boxers in the early stages before limbic degeneration, but progressive degeneration resulting in anterior thalamic and thalamic areas of the brain, resulting in dementia. Now, of course, trauma is multifocal. The brain is hit from so many different locations, in a boxer's career. So this is diffuse injuries, but it's interesting that despite the fact that it's diffuse, the limbic system seems to take the brunt. Herpes encephalitis. It particularly loves the hippocampus and the mesial temporal lobe. The same progression that you see here occurring in herpes encephalitis, with dementia being the result after a long time frame. Kernicterus, the same, a fewer cases, but the description was not dementia. It was described by the authors as retarded, idiotic, the old term descriptions for progressive loss of cognitive function in developing children. So this reaffirmed my initial feeling that limbic degeneration by itself, with other injuries on top, as might occur with boxing and herpes encephalitis, which may have a bilateral hemispheric injury pattern. But the stroke model is absolutely beautiful, because in those stroke cases, let me just go back here for a second. Some of these were strictly left-sided, some were strictly right-sided, and some were bilateral, occurring at the same time. And there were others that had a left-sided stroke at one period before they died, and a right-sided stroke at several years later. And what was astounding was the finding that the limbic degeneration associated with the older lesion had progressed farther in the tape circuit than in the more recent case, a stroke situation. Here's a graph that I did again about 30 years ago, showing the progression of dementia following boxing, long and drawn out. This is following stroke. This is following kernicterus, and this is encephalitis, much more rapid. You can see the onset, but nevertheless, dementia progressing gradually. So we've talked about localization, age of onset, sex distribution, and hemispheric distribution as well. I believe that the degree of limbic degeneration is directly related to survival time and the amount of time that it takes for the progression of the limbic anterograde transsynaptic degeneration to progress downstream. I presented this at the National Neurotrauma Society, where I met Anne McKee, who's one of the most foremost experts on chronic traumatic encephalopathy. And she reviewed my paper, and she suggested that this paper that I presented could explain a lot of the data in her studies. She did studies on quite a number of football players who died, and astoundingly, she had created her own staging, which I did not know until I read about in her paper. She called it stage 1, 2, 3, and 4. And here you could see the hippocampus and the mesial temporal lobe getting progressively more involved as a function of time in all of these different cases. And she wrote the article, Stage Progression of Atrophic Changes in Football and Blast Injury Related TBI with Increasing Hippocampal Atrophy Associated with Progressive CTE and Dementia. And these are some of the histological changes showing beta amyloid, neurofibrillary tangles, and all of the histological issues that Dr. Gonzales just presented. Now, I compared her stage 1, 2, 3, and 4 to my stages B, C, D, and E. And I applied the same technique, looking at her stage 1, 2, 3, and 4, and looking at her symptomatology and dementia And you could see the very same abnormalities in her histological studies on football related injuries. And so my stage progression as a function of time applies not only to stroke, but to the three other categories that I mentioned. Now, what about identifying limbic degeneration in a living patient? At the time that I wrote my paper and gave subsequent papers, I looked at the literature and I was astounded to find a paper in 2002 by Kodama and colleagues called Transneuronal Degeneration Patients with Temporal Lobe Epilepsy, Evaluation by MRI Imaging. And there we go. These patients had temporal lobectomies to cure their epilepsy that had resulted from temporal lobe sclerosis due to previous injuries to the hippocampus. And the radiologist identified radiologically atrophic fornices, mammillary bodies, and all the other regions of the brain that were described in my initial key case. And when I looked at the references, there was the original article that we published in 1977. And I was absolutely thrilled, honored, and fascinated by the fact that we might be able to identify limbic anterograde degeneration by neuroradiological approaches. And now we have MRI, we've got fMRI, we have PET scans, we have all kinds of other neuroradiological approaches that can be used. Looking at the various neuropsychiatric disorders and syndromes that I was familiar with in doing my residencies and internships, I'm going to suggest that many of the syndromes that we see in trauma and surgery, such as dementia pugilistica, the Kluver-Bucy syndrome following trauma and surgery, might be explained by limbic degeneration. The same with mesial temporal sclerosis, which we know, associated with epilepsy, can produce progressive behavioral and cognitive abnormalities. Post-encephalitic limbic dementia, as in herpes, neoplasms can produce the same kind of projection. Toxic metabolic. There are cases in the literature and in animal literature where dogs with rabies have been shown to have limbic anterograde transsynaptic degeneration. So we have animal models. And all of the limbic degenerations that I've described have also been shown in cats and rats and numerous other animals. So we now have multiple animal models that can be used to study this process. I also want to bring up environmental issues. There was some work in the veterinary literature where dogs back in the late 1980s and 90s were given bleached flour that contained an AGEN bleach whitening, flour whitening agent that caused a rabies-like behavior in dogs and had been given in dog biscuits to the dogs. When they did the autopsies on the dogs, they found that those dogs had hippocampal degeneration. The dog biscuit company went ahead and took out the bleached agent, and they were able to prove that agent was the precipitating factor in those animals. All right, so starting with my first publication in 1977, I've given talks at many other venues at the Neurotrauma Society Symposium in Santa Fe, where I met Dr. McKay, the Academy of Neurology Sports Concussion Conference in Denver, the US Congress of Psychiatry and Mental Health in San Diego, the Sarah Jane Foundation in New York. I gave a talk in Las Vegas to the Nevada Regional Neurosciences meeting where I was told to call my proposal a holistic approach to understanding the condition. And here we are today presenting the talk here. So the conclusion is that we need to collaborate together to find a way to prevent limbic degeneration from occurring. We can do that with boxing by creating helmets, perhaps, that can identify EEG abnormalities and also protect the brain from progressive injury, and taking a look at all the different lesions and proteins that occur in all these different diseases that can be associated with dementia, to see whether or not tau proteins, alpha synuclein, beta amyloid, et cetera, are the end results of the degenerative process, not necessarily the cause. And that is what I'm going to propose, and that we need to look at the new ways of looking at this limbic degeneration neurodiagnostically with neuropsychological test measures, neuropathological and neuroradiological markers of all kinds, biochemical markers, such as the ones that Dr. Gonzales was just talking about, neurophysiological measures, looking at prevention with prevention, helmets, diet, activity, elimination of the causes of hypertension and diabetes that we know can lead to higher instances of Alzheimer's disease, medications and vaccines. I'm bringing this up specifically now with regard to COVID. Just in reviewing the literature in the last 3 weeks, I found 20 papers illustrating that COVID specifically injures the hippocampus and the cerebellum and seems to have a liking for the pyramidal cells in those those parts of the brain. And then we need to look at behavioral treatments of a psychological, behavioral, or cognitive nature, neuropsychopharmacological agents that could understand the trophic and neurotransmitter abnormalities in those affected limbic pathways, and even look at electromagnetic stimulation and maybe a little far-fetched at this time, future embryonic stem cell treatments that could produce regeneration that would combat the degeneration that is clearly causing an Alzheimer's-like dementia in over 50 different diseases that we've identified. End of presentation, and I'm open for questions.
Thank you, Dr. Torch.
I'm sorry for taking so long, but I matched Dr. Gonzales's.
Great job. No, and do we have some questions from the group?
Yeah, I guess I'll start. So I'm interested in psychiatry, but I also have like a really strong appreciation for neurology. It's so fundamental. You mentioned in those last few slides about like cognitive behavioral therapies of sorts to help with this. When you do those therapies, have they done images or imaging studies or any other like marker studies to show changes after they take therapy, do therapy?
Well, you know, your question is great. Let's get back to the reason we're having this symposium. We all are educated and trained and professionally working in our own cocoon. There are psychiatrists who know about drugs, but they know very little about pathology. There are neurologists that know very little about psychiatry, let alone child versus adult psychiatry. There are neurochemists that don't know anything about clinical manifestations in a human being, but they are very well versed in animal work. Same for psychologists. We're all working in our cocoon. And I think the important take-home message of this symposium is that we need to bridge the collaboration between various researchers and clinicians across the spectrum. And if we could do that at the UNR campus and create an interdisciplinary dialogue that contributes to collaboration and a broader understanding and innovation, we will reach new highs. And I would suggest that the INDIGO mission should be the focus of the entire university, in specific to Alzheimer's disease. And I'm sure that mission can apply to many other situations as well. So my answer to you is get more training in neuropathology, take up some courses in psychiatry, sleep medicine, learn a little bit of neurochemistry and neurophysiology, and you'll be well set and on your way.
Sounds good. So Dr. Torch is convincing me to do a dual board certification in neurology and psychiatry. Those are competitive and they're hard. I'm joking, but no, I really appreciate that. I'll continue to look into this stuff. I mean, just as you were saying, I mean, it's hard to understand an issue when you're just like looking at it from one perspective. So I
Yes.
appreciate that.
And when you're doing your rotations in medical school, ask those same questions of all of your different the clinicians that you're working with. They probably won't have answers, but you'll be stimulating their thought processes and developing a pathway for yourself. When I started out as a young medical student, I wanted to know where the ego, the id, and the super ego were. I read all of Freud's work by the time I was around 15 years of age, and I was interested in trying to understand how the brain worked. And an old girlfriend of mine told me that I had confided in her that I thought the brain was made up of prisms and mirrors, and that light would enter the eyes, hitting prisms and being deflected in different angles to mirrors that went to different part of the brain, that went to the aha part of the brain where memory was stored and allowed for recognition of what you were seeing. Now that was quite an infantile, imaginative thought process for a young 12 or 13-year-old, but I decided to follow the rest of my path, looking at the various different means of study that have brought me to this position today.
Yeah, sounds good. That's prisms and stuff. That has like Pink Floyd vibes or something. I don't know. Anyways, yeah, thank you so much.
You're very welcome. Any other questions? Marissa, how about you? Oh, she's gone.
Blanking right now, but do you know of any studies that that have looked at like the question that soon to be Dr. Kim will be that he suggested? Do you know of any fMRI studies in patients who have undergone cognitive behavioral therapy or any of those other
No,
treatments?
no, I
Anything
have
you know?
not, but I think it would be wonderful to have psychologists looking at their therapies from the point of view of neuroradiological and neurophysiological studies. I knew very little of Dr. Gonzales's work. I mean, most of the stuff that he's taught us today is brand new and cutting edge. And one can't be an expert in all these different areas. And one really needs to produce a colloquium of collaborating people in different fields that can join forces together.
Dr. Gonzales, I wonder if you have a comment. I mean, for me, coming from the neurobiology, molecular neuroscience point of view, I'm curious your thoughts. You know, you've worked in labs affiliated with medical schools. From your perspective, from the non-clinical perspective, what are your thoughts on collaboration and how we can do better and get these technologies, get these advancements into the hands of clinicians and ultimately to patients?
That's a big question. I think, I mean, Dr. Torch brings up a very good point, is the need for collaborations and to spend, you know, get out of your comfort zone and really interact with people in different areas of science. And more importantly, I think people need to stop thinking that focusing on what they do is, you know, their world is their universe is the only thing that really matters. I mean, there's multiple multiverses, I guess, in science of how things are happening, but we never communicate until, you know, you bring us together in a forum like this, where we can, you know, a vascular person can talk to a neuron person, and then, you know, just expand the ideas more and more. I think it was really interesting bringing it back to vascular thing is that the hippocampus as a target is interesting because it has, if I remember right, a really high number of neurons, but a very low level of capillaries in there. And so blood flow within that region is highly efficient, but when you have something so finely tuned, it's easy to break down. And so it makes an interesting target for a lot of these, the beginnings of a lot of these diseases. Sorry, I was muted.
Sure. No, I agree, and I think I think the medical school training program should host a variety of researchers that can communicate with one another and to demonstrate to students the importance of collaborating and not going into one's own narrow world. It's a multi-universe. And just to point out also, tape circuit that it was a very simplistic neurodegenerative pathway, really has there are many other pathways that that are going to the frontal regions above the corpus callosum as well as below. And then I didn't even talk about the amygdala and its projections. And we didn't talk about memory, and we didn't talk about child abuse. We've talked about boxing, we've talked about football, but now there is a lot of work showing that corticosteroids released into one's bloodstreams as a result of stress can cause hippocampal damage in newborns and infants. And so what are we're talking about epidemiological issues of poverty and anger issues and single moms taking care of babies without support, resulting in frustration and unwanted child abuse and neglect. So, one could take this to many different levels. And having been educated and self-trained in these different areas, I've become very realistic of what's necessary. And I just commend Mary for what she has done in creating this meeting forum.
This is a group effort, and I'm really pleased with the response and really appreciate all of you who have come out to be part of this. I want to say it's 1:15 now. Are there any final questions before we wrap up and look forward to the next conversation? Andrew, you look like you're about to say something. Are we ready to wrap for the week?
Yeah, I just wanted to say, like, I was chatting with Marissa just now, and it's been like, it was really cool, like, just hearing from people who take this work really seriously, Dr. Gonzales, Dr. Torch, and just really applying yourself in this field. Me, I'm in medical school, and all I do is I always call med students um, like enthusiasts, because all we do is we just gather a bunch of information on a lot of stuff. That's kind of what school is all about. But applying that enthusiasm is like another beast. So I thought this was really cool, and I hope to follow up on not just, I guess, Alzheimer's disease, but I mean just all these things, as you're saying, Dr. Torch, like exploring different facets of medicine. Yeah.
Getting back to my days as a medical student, I got into the University of Rochester School of Medicine. I spent 2 years there, and all I did was memorize, and learning terms and terms and terms. And I said to myself, I want to be a doctor, but I want to be a researcher. And I didn't do very well in my first 2 years. And the dean became aware of the situation, and he invited me up for a conversation. He said, Bill, you have to get off the pot and decide what you want to be. And he says, how would you like to take some time out? And I said, what do you mean? He said, why don't you go for your PhD? So I literally did. And I dropped out and I joined the graduate school. And I worked at the Center for Brain Metabolism and the lab of biochemistry. And I got my master's degree in neurochemistry. And I realized hey, I really like working with people. I don't like working in a laboratory with animals and prisms and biomembranes. I want to go back to medical training. I went back to the dean. He says, welcome back. And I did 6 years there, and I graduated with my MD with honors in research on top of it. So sometimes you have to create your own pathway. And that's the point. And don't be afraid to do that.
Cheers to that. Well, I want to thank everyone, and I hope that we'll continue the conversation. Those of you who are in our Slack channel, we can continue to ask questions and discuss this topic. And we will post, I don't know, Andrew, I don't think we have the next Indigo meeting ready to announce, but we will soon, and we'll be sure to email everyone. If you haven't added yourself to the interest poll, that's the best way to get the email updates on when the lectures will be happening. And thank you so much. I'm so grateful to Dr. Torch and Dr. Gonzales. These were fascinating, very stimulating conversations. And I think I'm going to keep percolating on them through the week for sure.
Hey, Mary, is it possible to share a link or something, like something so people can like sign themselves up? Is that possible?
For the Slack channel?
They could either do the Slack channel or
it's in the email. I'm not on my,
Oh.
I'm in my lab computer, so I don't have that super handy. I see Gary waving to us from the hospital. Thank you, clinician in action. Good job. But it's in the email, and I'll be sure to send out a follow-up when we have the next announcement for the next Zoom meeting, unless you have it handy, Andrew.
I do not. Sorry about that.
That's
But
okay. Folks, it's in there.
okay, perfect.
Thank you all so much again, and look forward to seeing you soon.
Thank you, Mary. Thanks for all the work beautiful work you've done.
Thank you.
Yes, thank you.